In this particular lecture, we will be having an overview on how fluid dynamics and human health these are connected. While it is quite apparent that human body has certain fluids and these fluids are important for functioning of the different systems, but the real connectivity between the dynamics of flows in terms of what we learn as the fundamentals of this subject and how that applies to human health, that needs a bit of more focused understanding. So, if you look into the real world scenario, on the right I have put a diagram which is arbitrarily taken from the internet, but you can all realize that it conceptually represents a very advanced engineering plant where you have a very complicated piping system, and this is bread and butter for any engineering process where you have pipes, where pipes will transport different materials from one location to the other for many processes or sometimes even for power generation or other purposes. Now, having taught the subject of fluid mechanics for many years, when I was in my early stages of doing research on related topics, my general understanding was that any engineering analyst who can analyze the flow and stresses in such complex pipes would be a reasonable master of the engineering fluid dynamics. But then when I started my learning of the human body pipes, by human body pipes we cannot generalize anything. So, you can see an anatomical view in the left, and these views are quite common, because even if you have not studied advanced life sciences, at least in school levels also an outline of such things are that outline is presented. So, if you see you have a major system in the human body which is the circulatory system, and why I am emphasizing this is because although fluid dynamics in human health does not mean only circulatory system, but circulatory system will be one of the important focus of what we are going to discuss as a part of this course. So, what we see is that the circulatory system is not in isolation like some arteries, veins, etc., but the circulatory system connects different vital organs, and the functioning of these organs, each of these is an individual system or a part of an individual system. So, all what it means is that the circulatory system, although orchestrated by the heart and the lung, we have many other organs, in fact all other organs whose lifeline depends on that. And if you see the variety of the so called pipes in the human body, you have the very large ones like large arteries, and you also have large veins, and it is quite well known that the difference, of course there are many differences, but one of the major things is that their transportation of pure and impure blood, and the other aspect is that we can feel the veins from the superficial location in the body, but arteries typically they will be deeper inside, although certain arteries can be sort of sensed from superficial probing. Now, when you have these large arteries and large veins, we would definitely have something which are smaller than those. So, those are small arteries and small veins, and then even the smaller ones which are called as arterioles and venules. So, these are like baby arteries and baby veins, and arterioles and venules are connected by a capillary network which are known as microcapillaries, and these are like micro channels inside the human body. So, coming all the way from the large arteries, veins which are large in dimension, typically you will have their core diameters easily ranging several millimeters, centimeter, but if you get down to the microcapillary, that is of the order of microns, that means 10 to the power minus 6 meter order. So, you have a hierarchy of all these, that means you have the ones which are quite large and the ones which are quite small, but they are all connected. So, one is influencing the other, and that makes the problem of understanding these flows a multi scale flow. That means you have a large scale and you have a small scale, and large scale and small scale are coupled, and this is one of the major aspects that has to be kept in mind. Now, fluid dynamics as a subject. So, if you look into the keywords that we are going to connect in this introductory discussion, one is fluid dynamics and another is human health. So, when we talk about fluid dynamics, those of you who have done any course on fluid dynamics, your mind will be occupied with certain equations. If you have done it at an elementary level without getting into anything advanced, at least Bernoulli's equation is something that all of you have studied at some point of time, typically starting from high school, and then from all the way from that to Navier-Stokes equation. So, you will have your mind filled up with equations, and when there are equations, equations have solutions, and common sense tells that a majority of those equations are either differential or integral, that means they depend on calculus. So, the question comes that was there anything as a subject of fluid dynamics before the advent of calculus, and this is very very important to understand, because fluid used to flow. The Harappan civilization had outstanding water management system. So, all these were long back, even much before the so called the advancement of western civilization came. So, something was there as a knowledge base which could drive the developments around that time. Now, that is a very broad area of how the subject developed historically. So, we can broadly divide it into the pre-calculus age and post-calculus age. So, in the pre-calculus age, the possibility was to do experiments, but we have to remember that in those days the modern instrumentations were not available. So, how would people do experiments? And then when we get into human body fluid mechanics, doing experiment on some machine is one thing, but doing experiment on human body to study human body fluid mechanics is, even nowadays people would find it very challenging, when so much medical imaging and other non-invasive techniques are there. So, in those days nobody would dare for that. So, then how would the subject develop? Very interestingly, the subject developed mostly out of imagination and experience. So, one of the landmarks in the modern advancement of the so called fluid dynamics for the physiological system came in the hands of Leonardo da Vinci, whom we know as a great painter and one of the major personalities in the so called Renaissance of which led to the advancement of knowledge to the modern era. So, in the left what you can see is some of the anatomical drawings of Leonardo da Vinci. So, there was no way by which by any imaging in those days people would know how the heart looks in the human body, how it is connected, and just by his imagination. And this is one of the major things that anybody studying any subject should recognize, that learning a subject needs certain tools, but progressing in a subject with creativity needs imagination, and a combination of these leads to a mastery of the subject, not just what is written in the book, but what could be imagined which is not written in the book. Now, how this imagination can come? If you talk with a child, the imagination comes through fantasy, but that is not a serious imagination. So, when the serious imagination comes, it is usually connecting other experience to address something which is not known. So, Leonardo da Vinci was a polymath, that means he was an expert in many things, and of course, we know that he was an expert in painting and drawing. The Mona Lisa, which is there in the Louvre Museum, is still the tourists' main attraction when they are visiting Paris. So, from such experience he had a great mastery of getting into the inner details of say river and say soil, because such an artist will very commonly draw the natural scenes where river, soil, all these things will come. So, it was by absolute imagination he could possibly relate the river flow with what is flowing inside the human body and soil with flesh. So, in that way, through extreme imagination, he was the first person without any proof could show in the drawings that the human heart is made of 4 chambers in an era when it was believed that human heart has 2 chambers, and this was also a belief at that time because he had no way to prove it. And then things followed from there, but very interestingly, so we all have these documented things of the Western Renaissance period and all, because these have been parts of literature, the history of science, the history of art, and people would recognize it. In the right you see an anatomical painting with Sanskrit medical annotation, and this is something just out of my own curiosity when I was looking into that whether there was something in the traditional knowledge of the Indian system was something which is related to all these, and we found that yes, there is something, but I do not have the knowledge to decode it. So, I have absolutely no idea of what is written, but at least from the drawings it is clear that it has a reasonably detailed depiction of the different major anatomical features, and we have to remember that this is much earlier as compared to what Leonardo da Vinci did. So, this was there in a medical text on anatomy in the tradition of Ayurveda. Now, how these things got lost? Possibly because like in those days in the Indian knowledge system, the main way in which knowledge was transferred by Gurukul, that means the gurus will pass on their knowledge to their disciples, but it was not so well documented, and in the process there is likely to be a loss of main knowledge. And then with many things there are debates like invasion of outside people, etc., many of these records may have got lost. Let us not get into those issues, but just to understand that whatever we understand as modern physiological fluid dynamics at its absolute cutting edge technology had its origin long time back in very early of the human civilization. So, Leonardo da Vinci of course made very interesting anatomical drawings, and I have tried to reproduce some of the drawings here, but these drawings were more or less conceptual, and roughly 100 years after that came William Harvey, who was the first known physician to describe completely and in detail the systemic circulation. I will talk about what is systemic circulation in the circulatory system terminology. Basically the circulation which circulates the blood throughout the different systems of the human body, and the properties of blood being pumped to the brain and the rest of the body by the heart. So, every this kind of innovation, or what I would say discovery, is hallmarked by something new. So, he was the first who showed that valves in the veins, which was discovered by his teacher Fabricius, permit the blood to flow only in the direction of the heart, and these act as lifelines of the circulatory system. So, all what it means is that just like we have the valve in the piping system which will control the direction of flow, there are valves in the veins which do that. So, that was one of the major contributions of him. The second contribution, he also postulated the existence of tiny capillaries between arteries and veins, which are the micro capillaries or micro channels in the human body. So, we talk about microfluidics as a subject, modern subject, and the terminology came in the late 90s and early 2000s, and see in 1661 these tiny capillaries which are there, these tiny capillaries were first pointed out. Now, with all these there were foundations, but the foundation was not quantitative. That means people had a qualitative idea of there is something like heart, it has certain chambers, it is pumping fluid, it is going through different parts of the human body systems, etc., etc., and there is a directional control through valves. All these were conceptually known, but the most remarkable quantitative finding, which nowadays of course is in elementary textbook in high school or junior college, is the so called Poisson equation, which relates to the pressure drop and flow rate in a circular pipe or tube. Now, he was also a physicist cum physician. So, in those days people who were knowledgeable had a very holistic knowledge, not like the modern system that someone has a very narrow knowledge and would not have a broad holistic view. So, that we now have to talk about interdisciplinary education policies, etc., to make them make people align like that, but in those days all the genius people were naturally aligned like that. So, we will find mathematicians also working on electrical sciences, fluid flow, etc. So, what he did is he developed an apparatus, and the schematic of this apparatus is there, and now it will look like very primitive, but you have to understand that in 1800s making such a setup was not a joke, and not only making the setup, a very accurate measurement of the pressure drop in a pipe and the flow rate, and relating the pressure drop with the flow rate for low speed flow, which is the so called laminar flow. So, his objective was not really to do that, he wanted to understand the blood flow pressure drop when the blood is flowing through a tiny capillary, but because that is very complex, and this is what is very very important for anyone advancing in any topic, that to get into the complexity first one has to start with the simplicity. We talk about many electronic inventions which have disrupted our day to day life, like for example the smart TVs, the smartphones, smart watches, etc. Now, if you look into the TVs in the modern era where you have those panels and all, it will be very hard for you to recognize that maybe 40 years back, when the first set of TVs TV sets were available to the Indian households, those were TVs based on valves. So, you were the valve based technology. So, very commonly we have seen like TV is not working, now someone is slapping on the TV and the TV starts working. I mean, you people have not seen this, and you will feel it very amusing, because now in the smart panel, if you slap the panel you know what will happen. So, the technology has completely changed, but had that TV not existed I do not think this TV would have come. So, it is all very important to understand and appreciate that had this Poiseuille's theory not been there, we would have not been in a position to have the modern advanced understanding of how blood flows through arteries and veins. Now, the Poiseuille's theory was quite good in terms of describing the flow of a liquid which is following the Newton's law of viscosity in a circular tube, but the tube is a rigid tube. But in reality the blood vessels in the human body are flexible, they are not rigid. So, any theory which is based on rigid tube is not likely to give very accurate analysis. It will give some analysis which is better than zero analysis, but it will not give the exact accurate picture. So, there came the importance of the work of Otto Frank, and if you look into, I have purposefully kept the dates. See, we always when we study history, and we were bothered so much about the subject of history that in our school that it still remains as an horror, that in which year what battle was fought, etc. But if you see the overall essence, the objective is not really in which year Otto Frank was born or in which year he passed away. See, every major advancement it took about a century to develop. So, he was the first one who brought into the analysis the fact that the walls of large elastic arteries, and these are very important because the major heart related problems are due to these, they contain elastic fibers formed out of something known as elastin. These arteries are distensible, that means they distend when the blood pressure rises during the systole and recoil when the blood pressure falls during the diastole. And what he tried to do, see, this is where always what I would say the so called primitive science was much more advanced than the modern science. Modern science has brute force tools, that means even if you do not understand anything, you have a machine which will give you the result, or you have a simulator which will give you the simulation outcome. It may be a black box, if you understand, okay, even if you do not understand, the result is there. In those days such black box items were not at all there. So, what they had to do, they had to appeal to basic physics or basic maths to get a picture, the essential physics being represented by that. So, what he did is he considered this like you have a compliant elastic material which can hold volumes of fluid of different amount, so depending on its volume. So, if you draw an electrical analogy, it is like a capacitor. Just like a capacitor stores its charge, it stores fluid, and the capacitance of the capacitor which dictates the amount of charge along with the voltage. So, here the capacitance is given by the volume which changes with time. So, just like you have the simple linear capacitance law Q equal to C into V, you can have an equivalent capacitance for such a system, and then your advantage is that whatever basic electrical circuit theory is known, that can be extrapolated to analyze such a system, and that is what conceptually Otto Frank tried to put forward. Of course, beyond Otto Frank the advancement was much more rapid, because came the modern instrumentation, and all these things came because of the industrial revolution, different forms of industrial revolution, like even what we are now witnessing is a sort of industrial revolution which came from electronics, communication and data sciences. We do not call it as a revolution per se, but it has transformed many things just like the early industrial revolution. So, the progress has been much faster, and people have then by that time calculus was well described, by Newton, Leibniz and several others, and that is how the subject from then onwards had a very balanced view. On one side you had experiments and on one side you have a mathematical foundation which was primarily given by calculus. So, now with all those we are in a position to analyze the physiological flows, but we should have a starting reference from which we will start. So, I will assume that most of the participants here you may not be like from the clinical or biological domain, maybe a few are there, but I would assume that most of you are from engineering or other this fundamental physics or mathematics, these type of domains. So, most of you have learned fluid mechanics from such engineering or basic science point of view or mathematics point of view. So, now if we have to migrate from that aspect of fluid mechanics to this aspect of fluid mechanics, what are the major differences that we need to recognize and possibly put these important points in one place and keep that in mind. So, what I have done is we have highlighted all these that I was talking about, the large arteries, large veins connecting smaller arteries, smaller veins and then capillaries. So, like a hierarchical structure, you have the artery, then a first order structure, then a second order structure, then a third order structure. In that way you have branching and more branching, and then at the end these have become so small that they can connect the micro capillaries. So, the large ones will have high speed flow because their diameters are large, and the smaller ones cannot have high speed flow because their diameters are small. So, typically the large ones will have high Reynolds number, which is talking about the ratio of inertia and the viscous forces, and the smaller ones will have low Reynolds number. So, typically the large ones will have turbulent flow, which is highly disordered random type of flow, and the smaller ones will have laminar, more orderly flow. You have unusual multiplicity of tube branching and hierarchical structure, that is what I have mentioned, that you have really very complex branching system which you do not have in a engineered pipe. Unusual wall properties. Wall properties means what? The blood vessel wall properties. So, blood vessel wall properties will have some elasticity, so you will have the modulus of elasticity. And why I say unusual? Because it will vary so much from one individual to the other. So, if you talk about say engineering material, say steel of a particular composition will have a particular property, mechanical property. Now, we cannot say that blood vessel wall even of a particular age or many other factors constant will have the same property, because it will depend on the different conditions of health and disease, you cannot generalize it. Unusual pulsatility, that means we all know that blood flow, because you have a expansion and contraction, so it is like a peristaltic pumping, and you have also a time dependent pressure variation. So, you have a pulsatile flow, but this pulsatile flow is not really something which is very common for everybody, and you can easily recognize this because all the pulses, the electrical pulses that get through our cardiac system, that is also having a different signature. That is why if all of us do ECG right now, we will get different signatures. So, all what it means is that you have a pulsation, but unlike mathematical ideality, you cannot just write it as simple as P equal to A sin omega t plus B cos omega t like that. I mean, of course, you can break it like that with large number of A's and large number of B's in the form of a Fourier series, but the coefficients will be very much individually varying. You have the complex rheology, which is very important. So, rheology is how the stress is related to strain or maybe the rate of strain. Majority of the body fluids will have rheology, which is not as simple as the Newton's law of viscosity. blood in particular, and we will see that how that is very important in terms of dictating what is happening inside the human body. And again no, that is not unique, because everybody's blood composition is, even if it is slight, if it is slight, but it is different. Under different health and disease conditions it is different. Sometimes some cells become more stiff during some disease conditions like sickle cell disease. So in that way it is possible to have very different rheological properties of the blood under different conditions for different persons under health, disease, etc. And all these hallmark personalized variations, and this is very very important, and in healthcare this is something which has been ignored for a very long time, ignored in a sense that people do not talk much about that, but now it has come to the surface that personalized or patient specific recommendations. So for every disease there is a treatment protocol, and doctors have the natural knowledge of following that protocol. But the question is, will or why should the same broad protocol be used for everybody, because everybody is fundamentally different as a human body physiological system. So that should be recognized, and how will it come? It cannot come until that science is revealed through personalized data analysis, otherwise that empirical thing will go on. I keep on attending many of the medical conferences to enhance some of my insights on what doctors are thinking about all these topics, and in one such conference there was one doctor who was giving a very popular talk and everybody liked it, that why the same paracetamol 650 for the fever for everybody, right. Because that is the early management of fever for everybody, any adult, until it is known that there is a specific origin of the fever which needs more and different treatment. So question is why the same for everybody? Simply because it is seen to work, but there should be, and for such a so called obvious thing like routine fever management it is that. So for more complex matters like cancer management or cardiovascular disease management, these things are even more serious. Now I was talking about the different systems and what we will emphasize in this particular course. So the first what I will do in this introductory talk is to give you an overview of what is going on in these systems, because as you realize from the title of the course that it will be talking about modeling and simulation, but modeling and simulation without understanding what is going on for which you are modeling is of no good. Then you will have only some equations which you will solve without relating that to what is physically going on. So this is just a recapitulation, because all of us have studied these things in school, but with due respect to how we all learnt in school, I do not think that it is something that we have retained very seriously until and unless we are working on these topics continuously. So if you see the heart and you see the 4 chambers, so the only thing that you have to keep in mind while interpreting what I am talking about here is that what you see in your left is right and what you are seeing in your right is left, because you have to just turn yourself to align with what you see here. So the heart, what it is trying to do? It is trying to have a management of separate management of oxygenated and deoxygenated blood, that is what it is doing. So oxygenated blood enters from the pulmonary vein into the left atrium. So the atrium is the chamber which is there in the upper part, and you have 2 such chambers, and you have the ventricle that is the lower, and you have 2 such ones. So if you think it like a box, you have left atrium, right atrium, and left ventricle, right ventricle. So you can imagine like a box with this 2 by 2 compartment. So from the pulmonary vein, and this is where the lung is so important. So what the lung will do, lung will have the natural exchange of oxygen which you have inhaled with that blood, so that the blood becomes oxygenated, and this oxygenated blood is the so called pure blood that you need for the functions of the different organs or systems. So, oxygenated blood, it enters to the left atrium and then it is transported from the left atrium to the left ventricle. So, you have certain valves. So, you can see the right side of the picture is talking about the left atrium at the top and left ventricle at the bottom, and you see some valves like the aortic valve for example. So, this valve what it is trying to do, it is ensuring an unidirectional flow from the atrium to the ventricle. Then the oxygenated blood is pumped to the aorta for the systemic and coronary systems. So, you have this from the ventricle you can see that there is a turn in the arrow which is going in the upper direction and it is going towards the aorta. So, what is the systemic and coronary system? So, systemic is the different systems of the body and coronary is the heart's own pumping. That means, heart for its own functioning requires some pumping and that is coronary. This is the difference between coronary and systemic. So, deoxygenated blood which is there in the other side, it enters from the vena cava to the right atrium which is in the left of your picture. So, this is basically that impure blood. Now, the deoxygenated blood is transported from the right atrium to the right ventricle which is down, and you have the very well known tricuspid valve, a particular valve which controls the movement of that deoxygenated blood from the upper to the lower, and this deoxygenated blood is transported from the right atrium to the right ventricle and it is pumped to the pulmonary artery. So, you can see that it has come in the left and then it is pumped to the pulmonary artery to oxygenize the red blood cells. So, what happens is that this is where the heart lung exchange is occurring. So, this deoxygenated blood is finding out a way to go to the lung where the oxygen will be exchanged with this blood. So, that artery is the so called pulmonary one. So, I talked about the cardiovascular, but it is very important that we have another very related type of flow which is cerebrovascular flow, and cerebrovascular flow is you can recognize from the name that you have a very important activity of our life is the supply of blood to the brain, and there are barriers to that, and this is known as blood brain barrier and a very important topic of research. So, when you have this supply of this pure blood to the brain, if there are resistances or blockages, then the brain may not be able to get the nutrition and the oxygen, and this is what is called as a stroke. So, some of us who are not trained medical experts tend to confuse between heart attack and stroke. So, heart attack or heart failure is related to the heart is not working, and this is where the stroke is where you have a barrier of the oxygen supply to the brain, and there may be a clot inside and all which is stopping the supply. So, they are related, but stroke is related to that brain activity, whereas heart attack is related to the heart activity. So, it is very important to understand the arterial connection which can be lifelines for the supply of the major blood supply to the of the major blood source to the brain, and one such there are many such arteries, their names are written here. I am not going to read out these names because then it will become a very boring life science class. So, the main one which is of fundamental importance is the carotid artery, and we will talk about how the carotid artery flow can be analyzed with the help of what the foundation items that we are going to learn as a part of this course. So, in the left what you see is that if you are doing a computational analysis in maybe one of the these regions of interest where that rectangular box is drawn in the right, you can get a detailed visualization of blood pressure, how it is fluctuating. The typically the red ones are the large and the green and the blue ones are the lower values. So, a doctor who wants to treat a particular patient, if this information is available, the doctor will have a easy visualization of what is potentially happening. This may not be absolutely correct, but this will give a visualization of at least subjectively the matter, and this can lead to a more scientific way of treating the patient rather than just sort of based on the traditional clinical practice. Pulmonary flows, as I talked about, it is one of the very complex the fluidic system that you get in the human body. I will talk about some of these aspects because you will have detailed lecture on how to model and simulate that. So, the foundation starts with our respiratory system. So, you have the nasal cavity, and through the nostril the air which is inhaled will go to the trachea, which is the windpipe, so to say, and from the trachea it will go to the bronchus, and typically the plural is bronchi, then bronchiole which are the smaller ones, and then the alveoli which is like the air sac. So, in that way what is happening is the air in a very strategic manner is getting redistributed over a large area, because large area means you can have very easy transfer to something where here the something is blood. So, blood also is distributed like a network through small blood vessels, and because it is distributed through a network like small blood vessels, and air is also distributed like a network through this airway system, through the air sac like the alveolar, and the alveoli has its own branches, so on. So, this facilitates the gas exchange, and when the gas exchange is occurring, so you have the gas and you have the liquid which is the blood here. So, this is like a two phase flow. So, multi-phase flow which is taught in fluid mechanics and CFD will have a direct application in such examples. And why it is very important, because the lung alveoli, they will have a layer of surfactant which will try to prevent the damage due to the high shear which is there in such flows. But under disease conditions, when the lung gets damaged, these surfactant layers would be disrupted, and then there will be emergent breathing trouble, which the people in the acute stages of COVID started feeling. So, that is the major reason of respiratory distress. If it is a chronic problem like COPD, you have the chronic respiratory distress, and if you have an acute problem like suddenly it is emerging, then that is acute respiratory distress. And to relieve that condition, you may need a surfactant therapy like typically steroid type of medicine, which will try to retrieve that surfactant layer. So, what will be the dosage of that medicine? Is it all required? Who will take this decision? In the COVID period, we found that because most of the doctors who were treating COVID patients did not have much of idea about such acute conditions. So, there was of course some broad guideline possibly given by WHO or other medical organizations, and they try to follow it. But as I told that every recommendation will not work for every person, because individuals are different. Sometimes a high blast oxygen therapy with high dose of this steroid surfactant replacement will work for someone, but for someone else with that, that person's lung is very weak and cannot sustain, immunity is not that good. So, one has to see that what will work and what will possibly not, and if we have a detailed fundamental understanding of what flow is occurring, then that will give you a perspective of many things like diffusion of oxygen and carbon dioxide inside the lung, transportation of oxygen and carbon dioxide to and fro from the body's tissues, and blood flow through pulmonary artery and vein, and how they are all connected, not that they are individual. So, all in all, this is a very very important topic, and you will have detailed deliberation on this topic as a part of this course. Urinary system, I have not just generalized it as a excretory system, because urinary system is a very specific component of the excretory system, and it has a specific function which is not just release of urine, but many other very important things. So, what we all know that the two kidneys, they are the lifelines of the what we say as purging out of the waste elements through the liquid form of urine. So, how does it happen? So, you have the waste materials accumulating in the blood. So, the plasma of the blood where these waste materials will be accumulated, that has to be filtered so that the waste materials can be rejected or ejected from the body. Typically urine is one such way by which it is done. So, to do that, it is exactly very similar of how the other part of the circulatory system will perform. So, you have the large renal artery, and then you have the branches, the segmental ones and the intralobular arteries and so on, and then if you see that it will get into finer and finer branches. So, in the top diagram you have the major blood vessels that provide the blood flow to the kidney, and their names are written. Now, when you go in the next figure, you see that the blood flow enters from the renal artery, then branches to the radial arteries, the names are there, and then to different arterioles. So, very much in the same concept as the general circulatory system, and then provide the blood for the nephron, which is the filtration unit where this plasma will be filtrated. So, and typically we have about 1 million of such nephron, and these are not regenerated. So, nephron damaged means it is damaged, and if you have nephron damaged, what it will do is it will not be able to do the filtration. So, toxic materials will accumulate inside the body, which will build up pressure in the system and make the heart and lung also not functioning. Water will accumulate at different parts of the body. So, it will give a complete failure of the other organs over time. So, it is very very important to understand the regulation of water and electrolyte balance that is fundamentally happening here, the renal blood flow and how the arterial pressure there is regulated, and arterial pressure around that place is very similar to the systemic pressure overall in the body. The vein pressure is much less, about maybe 3 to 4 5 millimeter mercury, but arterial is quite high. So, I have talked about some of the major systems where the physiological flows are important, but here very briefly we will talk about what is there in the system. So, you can have many body fluids. For example, we have saliva, we have sweat, we have urine, but blood is what is the most imperative one. So, blood, if you try to understand what is the overall constitution of blood, it is well known, but its role in the mechanics of blood flow is very complex. So, if you try to broadly look into what the blood contains a watery part which is like water, but it is not exactly same as water. It also physically looks like bit of yellowish, light yellowish, that is the plasma, the watery component of blood. How it differs from water is that it has other constituents like the plasma proteins for example, and several other constituents. The plasma proteins albumin, globulin, fibrinogen, these are very important for the functioning of the different systems, typically the kidney function and the liver function, and that is why you will see for kidney function and liver function tests the concentrations of these in the urine or the blood, these are analyzed. Now, if you leave away the plasma, you have the cells. You have three types of cells: red blood cell, white blood cell and platelet, and these have their important functions. The red blood cells in terms of fluid mechanics are the most common to analyze. Why? Because 85% of cells in the human body are red blood cells. So, they are everywhere, and even if you consider the blood's constitution, about it, I mean 45 may be on a bit higher side, but very commonly between 35 to 45% you have the percentage or equivalent fraction of red blood cells. In the pathology term, this is called as hematocrit, which is the volume fraction of the red blood cells in the blood. If you are getting a diagnostic test report, sometimes it reports it as PCV, packed cell volume. If you are doing a complete blood count test, this PCV value is given. So, if you are doing a complete blood count test, and this is very common, which is called a CBC test. Why it is done is because it is not very specific, like with that result you cannot tell that someone has a particular disease, but many diseases will feature in terms of some abnormal values in some of the parameters. For example, very high number of white blood cells will mean that possibly an infection, where the patient is fighting with infection, because WBC helps you to fight with infection. Very low number of platelets means that there could be some internal hemorrhage or there could be a blood clotting irregularity. So, all these things are very much important for the overall functioning. And for the white blood cells, the different types of white blood cells are also, their counts are very important, like eosinophil, basophil, neutrophil, lymphocyte and monocyte, and many of these are related to infections or other ailments. So, their percentage is also very important. So, this is called as differential count. So, you have a total count which is called as TC, which is RBC, WBC, platelet, and different types of WBC, this is called as DC, differential count. So, the complete blood count test gives both the TC and the DC. Now, in terms of fluid mechanics, usually WBC and platelet is not coming into the picture of analysis unless they have abnormalities in their numbers, but RBC yes, and RBC usually if you do not have high deformation, it will look like a biconcave disc, but they are extremely deformable. So, if you see this a narrow passage through which they are forced to flow, and you will recognize that where from such narrow passage can come, you may have a blood vessel in which you have deposits of plaque due to cholesterol. So, if you have such deposits, then you will have the wall very much thick, and at some place you have only narrow passage by which the blood has a chance to flow. So, in that extreme narrow passage, these RBCs will pass through, but they will deform, and they will deform quite severely, and in the microcapillaries, which are human body microchannels, they will deform very severely. So, this is an example of a particle based simulation method, which we will not cover as a part of this, because it requires more advanced topics which we do not have scope in this short course. So, you can make simulations which will give you a visual idea of what is happening with the blood as it is flowing through maybe a few micron restriction. So, individual red blood cells, they can also have different types of motion, like there is something which is called as tumbling, back and forth, and then you have a motion which is called as tank treading, which is more like you have a particular orientation in the flow. So, tumbling and tank treading, these are two very common types of motions, and there are very good simulations of these visualizations you can see in open source in YouTube. Now, tumbling to tank treading transition is very important, because then the ability of the system to endure a stress depends on the transition of this type of motion one from the other. So, it is quite important that that is analyzed, but it is not that easy, because the red blood cells will not be in isolation, and they will be interacting with not just other red blood cells, but also other elements in the human physiology system. Now, because these cells are there and they are not so simple, so their involvement in the blood will make the blood very peculiarly behaving. So, the cells, when they are deformed extremely, so that they get they become a part of the main flow, so to say, because they are not able to agglomerate at a certain place. So, they can move with the flow, and in a way it will change the viscosity, but the viscosity will be like a Newtonian fluid. But if that is not possible, then they can form local clusters, which is known as Rulex formation, and when they form the local clusters, because of these clusters the behavior is very much different from a Newtonian fluid. So, then different types of mathematical models are there to represent it. So, if someone asks that is blood a Newtonian or a non-Newtonian fluid, it is very difficult to answer. Of course, in general non-Newtonian, but under certain shear conditions it can still behave like a Newtonian fluid. So, you cannot just say. The other thing is that when you have a very narrow blood vessel, it is possible that because of extreme deformation, the blood cells will get lifted from the wall, and we will see that because of this lift from the wall what interesting thing can happen and how that can dramatically change the viscosity or the viscous behavior of the blood. So, overall the central message that we carry from here is that there is no universal behavior of blood, and that makes it very complicated. So, as I was talking about that if you have a very narrow channel, so narrow channel means typically you have of the order of microns, at least tens of microns. So, what is happening that the red blood cells are highly deformable, but the deformation is more when the channel is narrower, because narrower channel is giving a lot of strain, and because of that strain the deformation is more, and with this deformation there is a lift force, and you can see in this diagram that there is this red colored vector is showing a lift force which is due to the deformation. So, these green entities are particles, deformable particles, just simulators of the like a red blood cell. So, they will have a tendency to go away from the wall, but the shear in the flow, which is because of this velocity variation, parabolic as an example, this will try to bring this cell close to the wall. So, when you have these two forces balanced, there will be a location away from the wall where they will stay, and they will not come exactly fall on the wall. That means there is a thin layer which is separating the red blood cells from the wall, and this is called as a cell free layer. So, this cell free layer will form in a way that we have to imagine that in a fluid mechanics system the major origin of the stress when you have such a channel is because of the wall shear stress. So, if the cells are not at the wall, then the wall shear stress goes down, because the cells are major contributors to slowing down of fluid at the wall. So, when that happens, then apparently the blood will appear to be less viscous. So, as you are making the channel narrower and narrower, and what you can see here, for example, in this diagram, that if you look into the bottom right picture, as you are reducing the capillary tube diameter, this deformation is more severe and lift force is more severe. So, you have the reduction in the apparent viscosity. We call it apparent viscosity because it is not Newtonian fluid, so we cannot call it viscosity. So, that means you make the tube narrower, you anticipate what your intuition says that the blood should have more resistance against flow, but because of this unique phenomenon it is just reversing. So, that was first observed by two physicians, doctors Farhust and Lindquist in 1931, but at that time there was no advanced tools of mechanics to understand that. So, it took several years, or in fact almost like close to several decades to get a clue on how that is happening. Still there is not a very detailed understanding of how it happens, but at least this mechanism of this competing, the two competing forces induced by the deformation, that is there. Now, another aspect which we will not cover as a part of this course is that when the blood is flowing, it is a multi-physics problem, because although in the human body you do not have any applied electric field or magnetic field, induced electric or magnetic field can actually play a very big role. So, how that is happening? So, if you look into a blood vessel, if you look into the left picture, the blood vessel wall is not like a pipe wall. So, the blood vessel wall has a type of cell which is known as endothelial cell. This endothelial cell has many things. One of the important proteins that it has is a glycocalyx protein, and the glycocalyx protein has a negative charge. So, what happens is that the entire system is neutral, electrically neutral. So, if the glycocalyx has negative charge, the remaining of the system will have equal positive charge to make it totally neutral. Now, when you have the pumping induced by the heart, that positive charge is moving, excess positive charge, and it is creating an electric field. Now, when you have an electric field, this electric field, because of its movement, like its natural movement of a set of ions, it is just like electron movement, but instead electron it is ion in the physiological system. So, it will set up a current, and because that electrical current is occurring, it will induce its own magnetic field. So, you see how these electromagnetic effects are coming inside the physiological system, although no one is applying electric field and no one is applying magnetic field normally, unless you are doing a treatment with electric or magnetic field. Also, the red blood cells have their own charges. So, they can interact by Coulombic interaction between themselves and also with the wall of the blood vessel. So, what happens is that with all these, cumulatively that magnetic field is created, and you can make a calculation by using the Maxwell's equations to see what kind of magnetic field is induced. This magnetic force induces nitric oxide release from the endothelial cells, which is a physiological phenomenon which promotes vascular relaxation and increases the flow. See, human body is a great adjuster. So, whenever we have less flow or flow restricted, the body tries to adjust it. How? It tries to expand the blood vessel. So, it comes as a message from the brain from somewhere, maybe from the hypothalamus or somewhere. But what will trigger the message? Something which will trigger the message is the phenomenon that there is a nitric oxide release from the endothelial cells. So, then it may so happen that this can relax and it allow more blood to flow in a condition when the blood flow is restricted because of something. So, this mechanism is overall physiologically appreciated, but mathematically it is or what I would say as fluid mechanically, it is very poorly understood. So, in this particular course we are not going to talk about many of these things, but my objective would be to at least give you a feel of how a foundation knowledge on these subjects can make one solve very difficult problems, which in the clinical science is still a mystery. Now I talked about simulations, but I will talk about a simulation which is not a computer simulation, but it is also a simulation in a sense that a simulation which is based on a physical device. So inside human body many things are happening, but you cannot do experiments inside the human body at your will. So how will you understand it? So one possibility is that by some bioengineering tools you make systems, engineered systems which have some similarity with the physiological system. Accordingly, there are two important topics which have emerged of late. One is known as organ on a chip and another is human body on a chip. So all what it means is that whatever are the functionalities of different organs or maybe a connected system with the circulatory system connecting many organs, you try to make an engineered mimic of that. So you make structures and you connect them, and the advantage is that if you are able to make such a structure you can play with that. That means you change the flow, change so many things inside the body, you cannot make such trials. But on the flip side, the outcome that you get from there you cannot rely much, because it does not have the complexity of what is there in the human body, but with the modern advancement of 3D printing it is possible to make very complex structures, and with the help of many fluids and the functionalization techniques you can make biomimetic structures, and to a large extent it is possible to get some insight, although not exactly what is there in the human body, but this is called as body on a chip. And if such a system is there, the advantage is that you can make lot of trial and error on such a system, and in that way you can optimize certain conditions. For example, nowadays for many treatments you have the targeted therapy, that means you have say a chemotherapy which should target a particular say tumor. Now when it is targeting certain tumor, maybe there are markers so that it will go to that, but exact targeting is, it may not be possible. So it will create the adverse effect on other healthy cells, and this is the so called side effect of such treatment. So to minimize that you need to see two things. One is how the drug is sort of curing the disease and whether it is having adverse side effects. So typically the drug development is you do such trials first in the lab, and these labs are traditional life science labs. So in those petri dish and all those culture systems they will do, and then you straight away bring them to animal trial if the early results are good, and if animal trials are good then you go to human clinical trial. Lots of ethical and other issues are there. It is a very complex and very expensive process, because billion trillion dollars of business depend on the introduction of drug to a market. So in that way there are many situations when it is seen that you make a trial of a drug, maybe in the lab it is working well, maybe on the animal it is working well, but on the human it is not working well. So the reason is that the trial environment either in the lab or in the animal is not having reasonable similarity with the human system. So it will give you wastage of so many years of trial and effort and research. So if a biomimetic system could be developed, a model system, a simulated environment, then by combining that with the CFD type of simulation you can quickly come to a recommendation which with minimal animal trial can get into the human trials and then see how it works. So that is one side. The other side is that if you have such a system which you are making as an artificial physiological mimic, then you can use such a system to study the different forces that I talked about. For example, the lift force which is there, and the tumbling and the tank treading motion, and this is what we do very routinely as a part of research in my research group. So the whole idea is to try to make certain biomimetic systems and see how they function, but how they function is one thing, but how they respond to different needs. For example, if you have such a system and you have a targeted drug delivery, as I was talking about that in some cancer treatment it is needed. So, one of the modern emerging ways to do that is magnetic nanoparticle aided therapy. So, you have a magnetic nanoparticle with which you tag the drug molecules, and by some magnetic field you target that nanoparticle to that cluster of tumor or any other cells where the drug will be delivered. Now, usually these drugs are trialed in again the traditional experimental setups where you will not have the deformability or other properties of the human body. Now, because of that you will find, see the basic mechanics, you have the nanoparticles coming with the flow. Now, you have the transverse magnetic field. So, magnetic field will try to bring it in the transverse direction, and that is important, but you cannot apply large magnetic field because that has other catastrophic effect. So, you need an optimal magnetic field where it will be just adequate to proliferate the wall and get into that say disease site. So, how will you know that what? So, one way is to understand the balance of magnetic and viscous forces, not in such a qualitative manner, but do detailed simulation, maybe experimental study. This is one such experimental study that we did in our group, and you can see that when you have this, you have a uniform wall, but with the flow, pressure, etc., you will see that they are bulging in and out the wall, and the particles are therefore captured differently where they are bulged more or bulged less, and in that way whatever you are speculating as uniform wall tube will completely fail, and you will see that it is moving differently. So, if you can make such systems and maybe complement it with simulations, you have a more realistic outlook of how it may perform in the real clinical challenge, and that is one of the important things. So, with all these you have the perspective of why we need CFD for physiological flows. It is not possible to obtain the complete details of flow properties like pressure, velocity, shear stress, vascular resistance, etc. These are certain things which in the human body you cannot really, even whatever medical imaging you may have CT scan, you may have MRI, different advanced MRIs, but still you cannot get point to point detail variation. So, CFD can do that, and not only that, it is like in a computer. So, you can give certain inputs, you will see the outputs, but if you give that input to a living human being, it can be a catastrophe, the person may die. So, you have a lot of the parameters which you can synthetically make a trial and error. It can help the clinicians with patient specific treatment strategy. So, how? You can give a visualization of what is happening through maybe the velocity, pressure, etc. data and other form of data. So, the physician, if the physician is not an expert in CFD, will have a visual thing, and with that visualization the physician can direct a form of treatment or therapy. So, in that way it can be much more advanced as compared to that purely experience or empirical type of approach that mostly they are forced to follow. So, these are some examples, some use cases. I will not really talk about many details, because the idea is to show that how these simulations are helping. See, there are many blood vessels which have branches, and near the branch you have the unidirectionality of the flow disturbed. So, you have vortices formed and so on. So, how they are going to impact the overall blood flow, you can very easily see by such simulated data. Similarly, if this is an example of a renal artery, that means an artery that supplies the blood flow to the kidney, that if you have stenosis, that means you have some resistances or blockages, then how that is impacting the pressure and velocity. So, you can get a visual appeal of that. So, this is an example of deep lung alveolar mechanics, and this is also very important. I talked about this, and this is the area in which we are collaborating with Dr. Saha, who is the resource person from the University of Technology, Sydney. So, with the help of the students from the two groups, we have tried to address the problem of what is the shear stress distribution in this deep lung alveolar framework. So, you can see that on the left you see the structure which is there in the physiological system, and if you just zoom each part you will see that you get at the end this is known as acini, which is like the finest of these branches, and they have a typical sort of ratio, the number of these branches from one node. So, once that is known from human anatomical structure, what our students try to do is to develop a alveolar on a chip. That means you can see this is an experimental setup from our lab, where this entire thing was sort of mimicked in a microfluidic chip, and then once you make an experimental study on this you will get pressure, flow, etc., and you can map that with color codes, and if you do CFD simulations you also get something, and if you could corroborate these two you can establish or standardize such a system which could be used to understand the impact of, for example, this surfactant therapy on how the lung is stressed, or maybe certain disease how the lung is stressed. So, and the other aspect which I briefly talked about and we will talk about in much details in our subsequent lectures is the deformation of the blood vessels. So, unlike the pipes, standard pipes, you have these ones which are highly deforming, and the deformation is influencing the flow and flow is influencing the deformation. So, it is a two-way coupling between the structural mechanics of the wall and the fluid mechanics of what is flowing. So, this is called as a fluid structure interaction problem. So, these are some examples where such simulations can give the visual depiction of how wall shear stress and pressure can vary in the large arteries, and these kinds of visualizations are very, very important if they are provided to a doctor to understand what is going on. And so, that is in the human body side. On the other hand, you have also what we say as artificial devices. For example, you have blood pumps, artificial heart, prosthetic heart valves, these are very common when heart valve is not sort of working correctly, artificial lung, artificial kidneys dialysis, that we know that how to have a purifying system where the human body kidney that those nephrons are not working, artificial vessels and grafts and stents which are commonly required like for example the angioplasty to make sure that the blockages are not creating hindrance and the blood flow is regularized. So, when you make such devices and you want to apply on a human being, it is a great risk and it cannot really be generalized that it will work. So, if such device is designed with the patient specific conditions by studying the flow and deformation etc., and then such devices are designed and fabricated and put into practice, there is a much greater chance that such artificial systems when interfaced with the human body will work more effectively. So, this is where designing of patient specific assistive or implant devices is one of the very, very promising areas of use of CFD. I already talked about the this alveoli on a chip. So, the general broad area of this technology is called as lab on a chip. So, whatever we do in a large lab can be all miniaturized in the form of a small chip, where you can do many activities. For example, say you want to purify plasma, you want to separate plasma for certain conditions, you want to have the separation of platelets from the red blood cells, because you are needing platelet for certain purpose, sometimes for transfusion you need platelet rich blood, and you can have a sorter for separation of RBC and WBC etc. So, there are many purposes for which you may need to work with such a small scale system, and this is in fact a complete different area on its own, and its design will require also CFD. And in such a microfluidic system you can work with DNA, which is a very important genetic material, and to understand the genetic sequence of a DNA, sometimes a complementary DNA which is complement to that, because a DNA will have this sugar which is shown schematically, you have a phosphate group which is negative charge, which is very important mechanics wise, and you have 4 nitrogen containing bases A, T, C and G. So, these nitrogen containing bases will be binding with their complements. T will combine with A and C will combine with G, like hydrogen bonding. So, when it does so, that is how one strand of DNA gets connected with another strand. So, if you are interested to figure out whether a particular sequence is there in a DNA, because that is a genetic testing. So, to do that you can put a complement of that strand on the wall of a microchannel and make the DNA flow. If that complementary thing is there, it will capture that and bind. But to make it move or capture faster, I will try to run a movie, but I am not sure it will work here. It will definitely show in my computer, but I am not sure it will show here. Okay, so it is not showing here, but I will show this separately. So, you have what I am telling verbally what is there. So, you have a oscillatory flap which is this dotted thing, which is a flexible material. So, it is programmed to oscillate in a way that the DNA will be forced to come to the wall and more DNA is available. But at the same time, if all DNA falls on one wall site where maybe a target DNA which is called as DNA probe, which is complementary to what you want to investigate, if everything falls there, you may have other investigating stations where nothing will fall. So, to see that what best can be done, you can use multiple probes, and then you can program their amplitude, frequency, etc., and it is a fluid structure interaction problem, but a complete artificial system, not the human body system, and then you can optimize the movement of the DNA. This work we did in collaboration with Professor Mark Madu's group with the University of California at Irvine. I talked about this DNA, and one example is also you can work with cells. So, when you have a cell and a cell is there on a human body, say say you have endothelial cell. Now, when you have the cells and there may be a cell which is adhering to the wall, then how the cell is sitting on the wall, it talks about the health or diseased conditions. Like if we are weak, we are putting certain type of force on the ground, and if we are strong, we are putting a different force. Cells are also like that. So, to understand the force shear interaction between the cell and the surface, and these are very different for cancer cells and normal cells. So, there is a way to understand the health condition of the cell. So, we developed a technology called as traction force microscopy on a chip, which will give a visualization of how the stress is distributed on such a cell. So, fundamentally what is done is you have a deformable channel on which this cell you put, and there are beads which are embedded in the channel. As the fluid flow is occurring, the cell is getting deformed and the beads get displaced. So, you take an image of this displacement, and by an inverse Fourier transformation you convert this displacement to force, and that is what is shown in the diagram. So, my former PhD student Dr. Tamal Das, he did this work. Currently he is a faculty member at TIFR Hyderabad. So, now the whole idea is that this is a simulated environment. Now, the simulated environment you can make more and more complex, and where really the CFD can help a lot. So, you do not have an isolated channel or an isolated capillary. In the human body you have this complex network, and so if you have a multi-prong approach, that you bring the insight of that complex network into your analysis through CFD, and you make that your so called lab on a chip system, where you try to reproduce those structures. And currently with the advancement of fabrication at micro and nano scales and maybe 3D printing, 3D bioprinting, it is possible to use bio compatible materials and deformable materials to make such structure. So, overall if you see that we are talking about physiological flows where something is happening in the human body, we are not able to get deep into that understanding because inside human body we cannot perturb in the way we like. So, we are doing two things, in fact a few things, not just two things. Normally to understand that, what we do, we have very simple measurements like blood pressure, pulse, oxygen saturation, that is done. Now I am talking a bit from the clinical side, how is it helpful. There are certain things for which you need diagnostic tests to get the blood report or other reports, maybe ECG or CT scan, MRI, depending on the need. There are ways in which you can do the CFD simulations by taking some inputs from those and construct some patient specific cases and get some insight, and you can also make these so called lab on a chip or organ on a chip devices to understand some aspects. So, if you can combine all these, you can get a multimodal analysis. Every aspect is different, but you combine together, you get a deep understanding on what is going on in a particular physiological reference. So, you can use that for making clinical decisions. For example, you can make a AI driven system where the patient comes, the patient gives inputs on medical history, symptoms, etc., and simple decision making framework, not to come up directly jump with a conclusion whether the patient has some disease or not, but screening, that means whether the patient is at risk, at danger or not. If that is there, then you get into some measurements, basic measurements, and after that you see a vulnerability, for example oxygen saturation is low. So, then you get into some form of say diagnostic test, and then with all these you can create a database where these plus CFD done on these the overall simulation of the physiological flow will give another dimensionality to your understanding, which is very different from what a doctor will directly gather through the clinical examination or diagnostic test. So, this on the top of all those can give a very scientific recommendation to the doctor that what can perhaps be done as a clinical step, next clinical step. So, overall to summarize, dynamics of physiological flows, these have captured significant research attention even much before the advent of modern fluid dynamics tools, and complexity of blood flows, it makes it significantly more challenging as compared to understanding engineering flows. You can make bioengineered microfluidic networks coupled with simulations which can give a unique perspective of studying physiological fluid dynamics, and this cannot be derived from live human or animal subjects. CFD can provide detailed quantitative insights on physiological flows which can be enhanced by taking inputs from clinical investigations and medical imaging data. So, all what it means is that you can have a simulated environment where you have one set of inputs which can come from MRI, CT scan, ECG, etc., that is a direct input. Using that input you can construct a CFD case, and then when you get that CFD output, that is a secondary more detailed information that you do not get from that. So, this combination can give you a individual centric recommendation by combining physics based and data driven approach, where the data comes from the individual patient's simulations. So, with this broad perspective we can sort of close this first session, and this will therefore bring us to the perspective of what we would study next. So, we have got at least the reasons and motivation of learning this subject. So, we will now from the next topic we will try to get into the basic mathematical foundations related to the simulations of these systems. So, that we will do from the next lecture. Thank you very much.